Our next speaker will be Dr. Michael Houghton. Michael Houghton was born in the United Kingdom in 1949. He graduated in biology at the University of East Anglia in Norwich in 1973 and obtained a PhD in biochemistry at the King's College in London in 1977. He subsequently moved to the United States, where in 1982 he joined the California-based biotechnology firm Chiron Corporation. At Chiron, he worked on what was at the time known as non-A, non-B hepatitis, and for the first time succeeded in isolating novel viral sequences using exclusively molecular and immunological methods. Dr. Houghton left Chiron in 2007, and in 2009 joined the University of Alberta in Canada as professor of medical microbiology and immunology and director of the Li Ka-shing Applied Virology Institute. He is currently working on the development of an effective HCV vaccine.
Hello, my name is Michael Houghton. I work at the University of Alberta in Edmonton in Canada. And today I'm going to relate to you the work that my colleagues and I did to identify and isolate the hepatitis C virus in the 1980s. So the work I'm going to relate is mainly work that was performed between 1982 and 1989. And my work continues at the University of Alberta trying to develop a vaccine against hepatitis C. But today I'm going to focus on the work we did to identify it in the 1980s. So in 1982, I trained in England, and then I moved to a company called Chiron Corporation, set up by Dr. Rutter and Dr. Penhote, both of University of California. And after arriving in the fall of '82, I discussed various programs with Dr. Rutter and Dr. Diener and Dr. Valenzuela. And Dr. Diener introduced me to non-A, non-B hepatitis. And we realized that maybe some of the methods I had used in my work on interferon genes in England could be applied to non-A, non-B hepatitis with a view to identifying the causative agent. So I was a molecular biologist, and looking at what we had available to apply to non-A, non-B, there wasn't much. There were infected human and chimpanzee materials available from different parts of the world, but polymerase chain reaction had not yet been discovered, and it wasn't until the late '80s that it started to be applied to labs on a routine basis. No antigen or antibody had been defined for non-A, non-B hepatitis, no cell culture system, no high throughput sequencing was available like it is today. And so, in other words, we didn't have a molecular handle. The work started off with a collaboration with Dr. Tatsuo Miyamura of the Japanese NIH. He came over with non-A, non-B liver samples from human cadavers, and he worked in my lab for almost a year. He also brought over non-A, non-B infectious blood from Japanese patients. We found that the quality of RNA that we could extract from the post-mortem liver samples was very poor due to degradation in vivo, so we realized that we needed to work with a collaborator who had an animal model working. There were several groups in the world who had a chimpanzee model for non-A, non-B hepatitis. We approached Dr. Bradley at the CDC, who was in Phoenix, Arizona at the time. He was an exponent of the chimp model. I'm showing two examples of transmission of non-A, non-B hepatitis from humans to chimpanzees that in his lab. We asked him if he would be willing to collaborate and provide samples for our molecular biological approaches, and he agreed. The main analysis that we used at the time was a plus-minus hybridization approach. So basically, we cloned polyadenylated mRNA from infected livers. We converted it into cDNA first with reverse transcriptase, cloned the cDNA into bacterial colonies, transfer the colonies to filter paper, and one can duplicate those filters, and you end up with a set of identical prints of the bacterial colony libraries. And we hybridized highly radioactive cDNA, either from infected liver RNA or from control uninfected liver RNA. And we're looking for signals that hybridize with the infected liver, but not with the control liver cDNA. So a laborious approach, one that we applied to many millions of clones. And at the end of that exercise, we had identified a lot of genes upregulated by non-A, non-B hepatitis infection, but none of them that we could consider to be viral in origin, because they all hybridized to the chimpanzee and human genomic DNA. So they were all chromosomal derived. So we thought, well, we better have pedigreed samples, and we better have samples of higher titer if we were to stand a chance at identifying this elusive agent. So we did request to Dr. Bradley, and we co-funded his efforts to do this, to identify samples of higher infectivity titer. What Dr. Bradley did is to go back through his archives of chimpanzee samples from many chimps, and he found a chimpanzee, number 910. We're looking at the ALT profile over the course of years. ALT is an aminotransferase enzyme that is released by damaged hepatocytes as a result of the infection. And so what you're looking at is a very typical example of non-A, non-B in chimps and in humans, where you have fluctuating peaks of ALT and hepatitis. What Dr. Bradley did is to make pools from around the peaks of ALT over many years. He came up with a large plasma pool that he then titered in other chimpanzees. So by diluting and transmitting the diluent to chimpanzees looking for hepatitis, he was able to conclude that this plasma pool had an infectious titer of at least 1 million infectious doses per mil. Similarly, the liver from this animal, chimpanzee 910, had an infectivity titer of around 10 million infectious doses, chimp infectious doses per gram. So we now had some better materials, better pedigreed materials. We applied the hybridization approach that I mentioned to you earlier. Once again, found a lot of genes upregulated by non-A, non-B hepatitis, which were of interesting nature for sure. But once again, none that could be shown to be extrachromosomal and therefore unlikely to be of viral origin. During this time, my lab was applying lots of other methods to try and identify Hep C. There was a lot of literature suggesting it might be related to a known virus, for example, hepatitis B or A, yellow fever, or bovine viral diarrhea virus, a toga virus. So we used these known genomes as hybridization probes under low stringency hybridization conditions, but could never see a specific signal. And we know why now, because Hep C is a very unique virus. We did wonder in the mid-80s if Hep C was related to hepatitis delta virus. There was some evidence from the NIH suggesting that there were similarities, perhaps suggesting a sequence relationship. So my lab got involved with John Geron at the NIH, and we identified the genome of hepatitis D virus, otherwise known as delta virus, for the first time. And this is an electron micrograph performed by Dr. Jim O of the genome. You can see that this is a circular piece of RNA. If you treat the RNA with formaldehyde at high temperature, like Jim did, you can actually see this circular piece of RNA around 1700 nucleotides in length. It's single-stranded, it's covalently closed, and it's a remarkable genome to occur in mammalian cells because it's highly related to plant viroids and virusides. So it's an unusual virus element to find in mammals, which was an interesting story. But the key question is, could we use it to identify Hep C? Unfortunately not. My colleague, Dr. Wiener, hybridized the whole genome of delta 2 extracts from Dr. Bradley's non-A non-B infected livers and plasmas, and the spots you see are due to positive controls that we included. That the key area on these plots is A4. That's where Amy had extracts of the high titer plasma pool from 910, and you can see nothing, even under low stringency conditions shown on the right. So once again, we could conclude that Hep C, whatever it was not related to hepatitis D. We were always pursuing other avenues. We had a lot of effort in trying to culture the non-A non-B agent in various cell lines. We now know that it's very difficult to do even today. Dr. Wakita in 2005 found a specific strain, one specific strain, that he was able to grow fairly well in tissue culture, in cell culture, but all the other strains are very difficult to grow. So this has been a very difficult virus to work on. We looked for reverse transcriptase activity in case it was a retrovirus. We could not see any evidence for RT activity. We looked for high molecular weight viral genomes, be they RNA or DNA, on electrophoretic gels. Couldn't see anything, either RNA or DNA. We also spent a lot of time trying to produce monoclonal antibodies that histochemically would bind to non-A non-B infected liver sections, not to control sections. We got antibodies to, once again, upregulated gene products, but nothing that we could decide was truly against the virus. About this time, Drs. Young and Davis at Stanford had developed a sophisticated cDNA expression system. Using a bacteriophage, lambda GT11, this was a highly efficient cloning vector, and each piece of nucleic acid that was inserted into this bacteriophage could be persuaded to express its encoded product into protein. And so one could easily produce very large proteomic libraries. And what Young and Davis showed, and some people before them actually, that you could use antibodies, characterized antibodies to specific proteins to fish out clones that way. And the process by Drs. Young and Davis applied to lambda GT11 was a very efficient one. So this was something we got interested in. Earlier in the program, I and others had considered this a very risky approach for a number of reasons. One is it didn't always work, even when a good antibody was available. No antibodies had been identified for non-A non-B hepatitis up to that point. And the persistence of non-A non-B suggested that there might be a weak immune response to this virus. I went next door to discuss antibody screening with my colleague and friend, Dr. George Kuo. He was not working on Hep C at that time, but he was a very skilled scientist in immunology and similar technology. And I explained to him how probably antibodies were limiting in non-A non-B. He suggested that maybe the reason why antibodies have not been identified so far was because of limiting antigen concentrations. Typically, people use liver sections of infected livers as the target for antibody immunofluorescence assays. So George thought that it could be limiting antigen that was the problem, and he recommended that if we use cDNA expression, by definition, you could perhaps get high levels of antigen expressed via GT11. And with those high levels of antigen expressed, we might be able to identify antibodies for the first time, and of course, in so doing, identify viral clones. So this was a very persuasive argument from George. Around the same time, Dr. Bradley was discussing using the same approach, the expression approach, cDNA expression approach, with another group. And so he was also in favor of this and suggested we go forward trying it. He later supplied some of the chimp 910 plasma to the other group that he was working on, as well as to ourselves. Unfortunately, we didn't get the answer quickly using this approach. Started off making a GT11 library from polyadenylated RNA from 4 different infected chimpanzee livers from Dr. Bradley. Dr. Kuo looked at various methods of detection and decided that the use of I-125 labeled anti-human Ig was able to achieve maximal sensitivity of detection. And then Dr. Chu screened this complex library using various convalescent sera from chimpanzees and patients. We all thought that using sera from individuals that had undergone acute hepatitis but then had resolved the hepatitis spontaneously without the use of antivirals, that would indicate perhaps that those individuals had the highest immune response, including antibody response. So we tended to use convalescent sera throughout this methodology. However, we obtained no viral clones, and then we switched to chimp 910 plasma pool, and once again, we could not identify any viral clones. So it was a frustrating time. At this point, we took a time out, so to speak, and we wanted to validate the whole cDNA expression system using hepatitis D virus. As I explained earlier, my lab had identified the hepatitis D genome, had identified antibodies to the virus, and so we had a supply of plasma with the virus in it, and we had a supply of patient sera with proven antibodies to the agent. So Kangsheng Wang and myself used HDV monkey plasma as a cloning source, excuse me, and we used HDV infected patient sera as a known source of antibodies, put the two together, and it worked very well. We got hundreds and hundreds of positive clones, and when we sequenced them, they were all hepatitis delta, virtually all hepatitis delta. So that was good news and reinforced our approach, but the delta monkey plasma infectivity titer was at least 5 logs higher than the non-A non-B chimp plasma 910. So that's 100,000-fold difference in infectious titer. Extrapolating the number of clones we got for delta to the number of clones we might get with delta, I came to the conclusion that, well, it's on the borderline, but we may be able to get this to work. So I returned to the non-A non-B plasma. I made a second cDNA lambda GT11 library from the plasma pool 910. This time we did do something different. Dr. Chu and myself, we chose to screen using a serum from a US patient who had abnormally high ALT levels. Normally, ALT levels in a chronic patient are in the 100 to 150 range. This patient, for some reason, had ALT levels in the 300s, 400s persistently. So it was an unusually strong hepatitis in this person. And we thought, well, maybe that indicates higher replication. Maybe this might be a better source of viral antibodies. And indeed, later on, after identifying Hep C, we did show that chronic patients have higher antibody titers than patients that have gone through an acute resolving phase of infection. But at this point, we were trying to think of anything different that we could try to identify this virus. And success at last. This happened around 6 years after we started in 1982. And I took the chimpanzee plasma pool, diluted it, ultracentrifuge such that the smallest known virus to man would be pelleted. I extracted total RNA and total DNA from that pellet, converted it into DNA using reverse transcriptase that we could clone into GT11, and then Quilim incubated serum from that patient with strong hepatitis with all the proteomics library from this plasma. And we were able to show that it truly was, if I go back to this slide, we got some false positives, as you would expect with this method. We got one clone, 511. It was only 100 base pairs. And this library that I'm talking about was the only library made from plasma that actually had viral clones in it. Out of 6 or 7 libraries that I made, this was the only one that had hepatitis C clones in it. And out of all of those libraries, we only got 1 clone by this immunoscreening method. And as I said, it was 100 base pairs in length, which is very small. So we were able to get overlapping clones, overlapping 511. We were able to show that for the first time in all of our work, this clone was not derived from the chimpanzee or human genome shown on southern blots. We showed that it was derived from RNA from infected liver, but you could not detect its binding to control livers, shown here. The virus contained RNA. It was sensitive to ribonuclease, not to deoxyribonuclease, and it was single-stranded. If you made single-strand hybridization probes, only one of the strands hybridized, shown here. The other strand did not. This proved that it was actually positive-stranded in the sense that the RNA encoding 511 was the actual RNA in the virus, what we call a positive-stranded RNA virus. Also, the RNA was very large. This is polyadenylated RNA extracted from the chimp liver, run on a northern blot, and this top band here is around 10,000 nucleotides. So we were dealing with a large RNA genome, positive-stranded. We then asked Dr. Bradley to send us acute hepatitis samples from chimpanzees that he had infected with non-A, non-B, or with hepatitis B, or with hepatitis A. And of the 4 non-A, non-B animals, we saw seroconversion to 511 in all 4 non-A, non-B chimps. We did not see it in 7 animals infected with Hep B or Hep A. This was a very important finding, clearly showing that these antibodies to 511 developed only after infection with non-A, non-B. We then obtained additional proof for non-A, non-B etiology. We had 11 patients from Dr. Gary Gitnick, clinically diagnosed non-A, non-B cases. George developed a 511 immunoassay, radioimmunoassay at this point, showed that 7 out of 11 were positive versus 0 out of 10 in controls. We were also getting more and more sequence from this viral RNA as we were walking the genome, as it's called, and we showed that it had a single large open reading frame. When we looked at the sequence very carefully, we could see very distant sequence identity with flaviviruses like dengue. It was very, very small distance homology, but nonetheless, it was present enough to indicate this was a flavy-like agent. And so now, in the fall of '87, we believe that 511 was derived from a non-A, non-B hepatitis viral genome. The proof of the pudding is in the eating. Then we had to, and obviously very willing to collaborate with groups around the world. One of the first groups was the one at the lab of Dr. Harvey Alter, and he had made a panel of sera containing either proven infectious non-A, non-B sera, infectious for chimps, or also containing proven negative samples that did not transmit to chimpanzees. And when George used the radioimmunoassay to assay this panel, for the first time ever, this panel was correctly cracked, decoded. 6 of the 7 proven infectious samples we could detect. As you can see, we've got counts in the many tens of thousands in all except for 1 sample, this one. So 6 of the 7 were scored positive. The pedigreed normal controls were all negative. This was background level in this assay, as were disease controls. So for the first time ever, we were able to break Harvey's code, which was very good news. We then looked at post-transfusion cases from him. He gave us 10 cases of post-transfusion non-A, non-B hepatitis. We were able to detect 511 in all 10. And he also provided us with the donors implicated in those post-transfusion cases. And in 9 out of 10 cases, we were able to identify at least 1 positive donor. We then went on and looked at other US non-A, non-B cases from Drs. Gitnick, Redeker, Dienstag, Stevens, and Tepmeyer. And of 24 samples, we scored 17 as positive, which was 71%, versus a background of 0.5% in healthy blood donors in the US. And finally, we scored 58% of Dr. Miriam Alter's sporadic non-A non-B cases as positive in our assay. We then turned worldwide, and Doctors Benino and Colombo gave us 32 samples of chronic post-transfusion non-A non-B hepatitis cases from Italy. 84% were positive. And then we got a panel of chronic post-transfusion non-A non-B cases from Doctors Katayama and Miyamura from Japan. And we found 78% of those positive in our assay. Interestingly, Doctors Katayama and Miyamura also sent us 13 acute resolving cases of post-transfusion non-A non-B hepatitis, and we detected only 2 of those, 2 out of 13, indicating 15%. And as I said earlier, it turns out acute resolving patients have less antibody titers than chronically infected cases. So our search for the virus ended in 1989. After 7 years with the publications in Science. We had back-to-back publications. The first one showing evidence that we really had identified a cDNA clone from blood-borne non-A non-B hepatitis. I've mentioned all the names to you already, except Dr. Overby. He was a hepatitis expert who really formed the infectious disease diagnostic division at Abbott Laboratories, and he joined us in 1983 or 84, I can't remember exactly. But he was a great source of information about viral hepatitis in general. He was a great mentor and a great supporter of our work, so we are indebted to him. And then the second paper back-to-back was the one from Dr. George Kuo and Hui-Lim Chu, Harvey Alter, and all of our collaborators, and obviously Dr. Bradley, where we looked at different human samples from all over the world and showed that indeed Hep C was the major blood-borne non-A non-B hepatitis virus. We walked the genome until we got to the ends, and it's about 10,000 nucleotides long, the viral RNA. It encodes for a lot of different proteins. It actually encodes for one single large polyprotein precursor that is cleaved through viral enzymes as well as host enzymes into a number of different proteins. And this was really, I suppose, the birth of the hepatitis C field. Now academics, industry, and public health could get involved in using this information of the genome to develop diagnostics. We had already got one with 511. 511, by the way, was derived from NS4 protein, a non-structural protein 4A. As we walked the genome, we could identify many other B-cell epitopes elsewhere in the viral proteins. So over time, we delivered more and more sensitive antibody diagnostics. The 5' untranslated region of the viral genome has a very precise secondary and tertiary structure to which ribozymes bind to initiate translation of the protein. It turns out my colleague Dr. Han showed that this is a highly conserved region of the genome and therefore is exquisitely suitable for PCR assays and TMA assays to detect viral RNA in samples. So over time we developed very good genome nucleic acid specific tests as well as the antibody tests. Many groups got involved in identifying the replicase encoded in NS5B. NS5A became a major antiviral target from scientists at Bristol Myers Squibb. This protein is involved in virus assembly and secretion, and it's very important. And scientists there developed very good antivirals against it. And many groups working together identified an inhibitor of the 5B polymerase, including Dr. Michael Sophia. In the NS3 region is a protease, a serine-like protease that has formed the basis for generation of specific drugs that have been used in humans very effectively. And then at the N-terminal region of this polyprotein, we see the two envelope proteins the nuclear capsid protein. And this information has led us, and my group still working on it in the University of Alberta, to develop vaccines based on the recombinant envelope proteins, as well as T-cell epitopes throughout the non-structural region. So the genome has provided the platform for the development of diagnostics, therapeutics, and vaccine candidates. It remains only for me to say, once again, that the identification and isolation of this virus was a team effort. And I really would like to acknowledge the contributions of Dr. Chu, Dr. Kuo, Dr. Bradley, without whom I would not be talking to you today. Thanks again very much to the Nobel Committee for this honor and for honoring the hepatitis C virus field. We're very grateful. Thank you.